Okay, so I want to change gears a little bit, and instead of thinking about the basal ganglia, I'll have us thinking about the cortical system of eye movements and attention. But we'll try to stay in the same questions of information sampling, and we'll look at them from the perspective, starting from the perspective of reinforcement learning. As we studied this topic of information sampling in the lab, it became clear to us that it's useful to divide it into two contexts, at least for the purposes of study. One is the case when you sample information in the context of the task, so in order to guide a specific action, when such as what I spoke about this morning. When you're preparing a cup of tea, you know your task. You also know you have learned an efficient sampling strategy, and each one of your eye movements has a purpose towards furthering the goal that you're trying to achieve in the task. So this is the scenario that I will talk about today. Tomorrow I'll give another talk, and the title of that is Sampling Useless Information. And for that talk, what I'm referring to is when you sample information outside of a task context. Or apparently, at least the experimenter doesn't really know what you're doing, so you're sampling, you're serving some internal goal, right? And that second kind of sampling is actually more difficult to tackle because it's more difficult to know what the internal goals are. So tomorrow I'll talk about exploring some ways to get to that part. But for today, let's stay in the more perhaps simpler paradigm of we're trying to achieve a goal, and on the way to that goal we carry a sequence of actions, and information sampling is naturally woven into this sequence of actions, right? So as we all learn how to cross the street, and it takes quite a bit of learning, we seamlessly combine these, right? So we stop before crossing because we know there is uncertainty at that juncture. We sample information, we look at the traffic light, we listen, and based on that sampling we decide what to do, and then we get a reward with either success or failure. So we also know, as I spoke and as I mentioned this morning, as we can really appreciate, that eye movements, rapid eye movements, saccades, are a prime means by which we sample visual information. And we know that saccades are deployed in this purposive way during the task, but what we don't know is how. How does the brain generate those signals to select the correct target that will best guide our actions? So let's think a little bit, because we want to stay in the realm of reinforcement learning. How do we represent this decision chain, right? We might represent it in a model as a Markov decision process. So let's say that I'm at this point, I arrive to an intersection, and I have to make a choice. What will I sample, right? So let's say I decide to look at the traffic. And then I can observe either there is a car there or not, right? And that observation will tell me the probability of the most desirable action, right? If I see a car, I should stop and not go, right? So that tells me that my probability to get a reward is 1 if I stop. My probability to get a reward is 0 if I go, right? And in either case, whether the observation tells me to stop or to go, sorry, I'm getting ahead of myself. In either case, I'm getting a high probability of reward. But say, on the other hand, that I make the decision to look up at the cloud. Why not? It's a legitimate target. Then I can observe the color of the cloud, whether the cloud is white or gray. But that won't tell me anything about whether I should stop or go. So let's say here, for simplicity, let's say my prior probability is 50%. That cloud observation will not change that prior probability. And so, in either case, whether I decide to stop or to go, my probability of reward is only 50%. It's lower. Okay, so the question that we're trying to answer is, we're trying to look at this stage, right? So we need some mechanism that will assign value differently to the traffic than to the cloud. And notice that this stage is prior to the actual sensory input, right? I can make this decision with my eyes closed, based only on my knowledge of of what these two stimuli are telling me, right? And this advanced decision of what to sample is the critical decision, because it will determine what it is that I'm looking for, what it is that I'm putting my eye on. Okay, so this is that's what we understand. How does the brain assign different value to these two stimuli for guiding the eye movements? Okay, so what possibilities do we have looking at this model? One possibility I already mentioned, and that is the probability of an outcome, right? So we can learn through experience or through explicit teaching, in this case, that if we take the sequence of action of looking at the traffic and then making a decision, we are much likely to get a positive outcome, to cross the street safely. Then if we take the sequence of action of looking at the cloud and then making a decision, that has a much lower probability of reward. And we also know, of course, in reinforcement learning, you can take that outcome that comes at the end of a sequence and back it through the decision steps, and so you can assign different value to the sampling decision, which comes earlier in the chain. That's one possibility. And another possibility is that you can see that there's another thing that distinguishes these two stimuli, in that one, the traffic reduces the uncertainty in your decision, so it gives you policy information, right? If you had two equally likely actions a priori, the observation of the traffic has reduced this to one, so has really reduced your uncertainty about which action to take, whereas the cloud does not. So this is another factor. The brain could actually compute the reduction in uncertainty or the policy information that is associated with each stimulus, and that may be different from the or additional to expected reward. All right, so this is the question that we'll examine. So before I go on into the experiment, let me introduce you a little bit to the pathways for generating saccades. So we know that visual information comes, this is a lateral view of the monkey brain. Primary visual cortex is in the back of the brain, V1. We know that visual information comes in there. It gets relayed through several visual areas, including V4 and the medial temporal area, that analyze visual features. And eventually the signals come to two areas that we know are important for eye movements. One is the lateral intraparietal area, LIP, in the parietal cortex. The other is the frontal eye field, in the frontal lobe. These areas are strongly interconnected with each other, and they send descending projections to the superior colliculus, which eventually drives brainstem oculomotor nuclei and eventually moves the eye. So areas LIP and FEF are particularly interesting because we think that they make the decision about where to make the next eye movement. They select stimuli for the next eye movement. Here we worked primarily in LIP. Okay, so just to give you an overview of the neurons that we're targeting in LIP, these are neurons that have spatial receptive fields, right? So every cell encodes a location in visual space, and the neurons, they receive information from lower visual areas that are selective to stimulus feature, but we think that in LIP the feature information, it gets sort of scrambled. So the neurons mainly tell you about space. They really encode a vector, and it's relative to the center of gaze. And another important property that they have is that the neurons don't respond to just anything that enters their receptive field, right? So when we move our eyes across a visual scene, we're constantly, our retina presumably constantly receives inputs. But a lot of these inputs simply do not show up in LIP. So the neurons have this magical property of silencing most things that are not relevant at the moment and pulling out objects that are likely to attract attention, that are either salient or task relevant. And so sometimes this is called a salience map. Okay, so this is from an older study by Itti and Koch. So if you have a complex visual scene, such as that one, and you imagine its representation in LIP, where every neuron represents a location in space, you can see that most of the neurons in this case are relatively silent. There is a bright locus of activity down there in the middle bottom, corresponding to that bag in the picture, right? And that tells you there's something interesting at that location in your visual scene, and that interestingness could come either because of feature contrast. The bag is white and stands out visually relative to its surround, or it could come because of a learned relevance. So in this case, the bag has $sign, so we have learned the significance of that. Okay, so this is what we know about LIP neurons. And there's also evidence that these neurons have two types of effects. They project forward to the front light fields and superior colliculus, and then they bias the brain to make a saccade toward that selected location. And there are also feedback effects onto visual areas, which can produce attentional modulations in those areas. So even if you do not move your eye, you can still enhance the neural responses emanating from that location, and you can better discriminate those stimuli. Okay, so this is a pretty powerful central selection that just says, here's what's interesting. You can look at it or covertly attend to it, but somehow the decision has been made there. Okay, so this is, as far as we know now, the selection signal is there. It has all kinds of effects. The question that we know less about is, how does it come to be, right? How does LIP know which stimulus to select? Okay, so we have what goes into this box? Okay, so we have the beginning of an answer from experiments that tell us that the target selection response in LIP is sensitive to primary rewards. And this is done in experiments where, for example, you take a monkey and you put him in a lab and you show him two stimuli. Let's say one is red and one is green. And the monkey learns that by making an eye movement to the red stimulus, he has a large reward, and making an eye movement to the green stimulus, he has a small reward. And then you see that the LIP response, when the monkey selects the red stimulus, is much larger. And when he selects the less rewarded stimulus, is much smaller. So the expected reward really shapes that target selection response. Okay, so these are very robust modulations. They're replicated by now in many, many labs, and they happen in a lot of neurons in our hands as well. So it's undeniable that they're there. But there's some questions about it, right? Because I've just shown you that in natural behavior, nobody gives you primary rewards for making eye movements. Eye movements indirectly serve the goal by sampling information, guiding an action, which eventually leads to a reward, right? So how does the reward act really come into play in natural behaviors? And the related question is, well, if the primary purpose of moving the eye is to sample information, how does expected information come into play? Okay. All right, so to get at this question then, we started from this intuition of natural behavior where you have information sampling. You sample information, then you make a decision, then you experience an outcome. So we wanted to create a similar condition in the laboratory for our monkeys. So here's how the task went. The monkey began by, a fixation point appeared on the screen, and the monkey began by looking at the central fixation. And then he saw a display containing 3 stimuli. At the end of the trial, we'll require the monkey to make a saccade, to make a decision whether to move upward to the upward white dot or to the lower dot, right? And in the beginning of the trial, he doesn't know which of the 2 dots is correct on that trial. But before he makes the final decision, we train the monkey to make the first saccade to this round patch, right? And when he makes the eye movement to that patch, he gets information in the form of coherently moving dots. So let's do it again. The patch appears, the monkey is preparing to make an eye movement. When he makes the eye movement, that triggers the information, so the dots in the patch begin to flow. If the dots flow downward, that tells the monkey that the lower target is correct on this trial, and the monkey is free to make his second saccade to that target. If the dots flow upward, it tells the monkey that the upper target is correct, and then he'll be rewarded for that target. Okay, so we are interested in the responses preceding the first saccade. That is the saccade that samples the information. Okay, and remember that I told you that we want to understand how stimuli are selected based on their informativeness, right? Based on this abstract property that you have before actually discriminating the motion. So so this stage where the monkey is sitting and observing the patch is critical. And in this stage, we gave the monkey advanced information about the validity of the cues. And we gave that information by means of the color of the border around the cue. And we had, so the cue that had a black border like this, the monkey knew it was a perfectly reliable cue. Whenever it said that the rewarded target is down, that was correct, and vice versa. But another cue had a green border, and that had only 80% validity. So that cue was simply unreliable. Sometimes in 20% of the trial, it said go to the lower target, but the reward was in fact at the upper target. And then we had another cue that had a blue border, and that was 55% validity, so slightly above chance. These cues were highly familiar, so the monkeys could, they knew their statistics and their reliabilities, and these remained constant across the experiment for the monkey. The two monkeys were tested. We scrambled the color validity mapping across monkeys. Okay, so the question that we're after is, will the neurons scale their selection signal according to the validity of the cue? Before I get into that, we have to see whether the monkeys understand the concept of validity. So we tested this using choice trials. These trials were exactly the same as what I've shown you, except that the monkey had two cues of unequal validity, and he was free to choose one. Right? So for example, if he moved his eye to the black cue, then that cue gave its motion information, and the monkey could finish the trial. And we had 3 pairs of these cues that differ in validity by different amounts. So for example, a difference of 0.2 means we pit the 100% valid cue versus the 80% valid cue, right? A difference of 0.45, we pit the 100% versus the 55%. And you can see that for the 2 monkeys, they choose the better, for all the pairs, they choose the better of the 2 cues on around 90% of trials. Yeah?
The optimal choice, I think you might consider the optimal choice is the more informative or like 100% valid?
Yes, the optimal choice is always the more valid one, yes. Yes, and so the monkeys get they get that, pretty much, yeah. Yeah.
Just to understand, you coach it all in terms of foraging for information, but in fact a reinforcement account.
Well, this is what we're going to get. This is what we're getting to. It is very similar, and I'm going to get to how we try to dissociate them. Right, right. Yeah.
Just out of curiosity, did you also look at the recording and the saccades for this? Because this information
during
the choice trial.
During the choice, yeah. So let me show you this, yeah. So during the choice trials, I'm going to show you now the neural responses. And we have to introduce the receptive field in this picture. So that's the dotted line there, right? So let's say you have 2 cues of 2 different validities. Let's say the white border is the better one. The monkey always makes his eye movement to the better one. But the better one is sometimes it's in the receptive field of the cell, sometimes it's opposite the receptive field, right? So the direction of the saccade will vary. The monkey's choice is relatively constant. Okay, and here the population responses, and I'm showing them to you we have different pairs of different validities, right? So let's walk through them. So if you look at the top pair, there is a 100% Q versus a 55% Q, right? So the monkey looks, so on some trials, the 100% Q is in the receptive field. So what you see, the neurons have a big visual response when the two stimuli appear, and then they maintain the sustained activity until the time of the eye movement, right? The time course here is the stimuli appear at time 0, and then we enforce a little delay period. We make the monkeys wait a little bit until we remove the fixation point and let them make the saccade. The saccade happens, it's cut off. It happens after 500 milliseconds. So this delay period is the critical period that people think that's when the neurons show you the decision in advance of the movement. So that's what we're focusing on. Okay, so if you have 100% Q in the receptive field, you have selected a target, you're planning a movement to the receptive field, the LIP activity is high, representing that location. In the blue trace, what you see is the 100% Q is opposite the receptive field. In the receptive field, you have a poor, a 55% Q that you're not choosing, right? Now the monkey makes the eye movement in the opposite location, so the activity of our neuron increases but then goes down, right? That's not its preferred direction, so it tells you, you're not making a saccade to my receptive field location. You're making a saccade away. So the selection signal is really the difference between those two curves. And the salient point here is that you see that this difference between the two curves, between the directional signal, decreases as the difference in validity between the stimuli decreases, right? So in other words, in all these cases, the saccade to the receptive field and opposite, they have identical metrics. The stimuli are similar, same luminance, same amplitude, same direction. But so the neurons tell you about the direction of the saccade, but not only about that. They tell you about the relative values of the options that are governing that saccade. Yeah.
Sorry, just to clarify, so the Q's can always incongruent, so whatever is in the most valid one is pointing one direction or the other?
They're always different in validity, yes.
Not in validity, in directionality, so they're always queuing opposite.
They're always opposite. No, I'm sorry, they're not, okay. They're not, we're looking at the saccade to the Q. So we put one of those, we put, in this,
right now they're not displaying any motion.
In this example, the receptive field would be here. So this is before the motion, and it's before this second saccade. We're not looking at this one. We're looking at the first saccade, right? Okay. Okay, all right, so the neurons are sensitive to validity. And now we have to get to the question, is it expected, could it be simply expected reward, or is there something else? Okay, all right, so let's go back to the MDP model, and let's try to write this, this task. So let's say you have a 100% valid Q, right? You see a black border. And then you observe a motion. Let's say, for simplicity, you see the motion going up. So you know that the probability you want to select the upward target at the end of your trial. You make a decision, you select the upward target, and you get a reward with high probability. And similarly, if you have you observe the green Q, you get a reward with only 80% probability. If you observe the blue Q, only 55% probability. Okay, so expected reward could explain everything. The action information is also a possible explanation. How do you tease those two apart? So we scratched our head about this for a long, long time, because in an action context, you sample information in order to improve your future rewards, right? If a signal is irrelevant to the task, you don't sample it. Right? So how are we going to distinguish between this? So finally, we got a little breakthrough in saying that, yes, information gain always leads to reward gain, but the opposite need not be true. In other words, you could have valuable actions that lead to reward, but they may not bring you new information. Right? So it's something like this. If you have a set of all rewarded actions, a set of actions that are informative and rewarding is only a subset of that. So this is our strategy to create. So I've shown you, we can look at our informative cues, right? That's a rewarded and informative stimulus. And let's create another condition in which the cues are rewarded but uninformative. Okay, how are we going to do that? So we chose to introduce a second step after delivering the information that the monkeys had to do in order to obtain the reward, but which didn't tell them anything new, right? So again, we have the same context of the monkeys observe the cues and they observe the motion. I just collapsed this into one step. And just as before, they make their decision and they get their outcome. But we introduce another step where after observing the cue, the monkey has to look at another stimulus that looks very similar to that cue. It's also a round patch. And only then they make their decision and get the reward, right? So they will look at this stimulus. It's also a patch that has dots, but this stimulus, when they look at it, it delivers only noise. Okay, so no new information here. Nevertheless, the stimulus is valuable because it's necessary to obtain the reward. Okay, and the same thing here. After they observe a green cue, they have to look at another patch that gives them only noise. And after they observe the blue cue, they have to look at another patch that gives them only noise. And we paired consistently the different color patches with the different validities so that their long-term reward expectations were the same, were equated. Okay. So this is where I'm having a lot of problems, and people say, well, but you know, these uninformative things, they're not really rewarding because this and that. The point is that they are really rewarding. The monkeys are looking at them. They wouldn't be looking at them if they weren't rewarding. And so I had to enlist Manuel's credentials as his modeling credentials to do a little Q model simulation and show that indeed, if you're computing Q values, state action values in this decision chain, the action values will differ here for the valid cues, but they will also differ here for the uninformative cues, right? Simply because these cues lead to a future reward. And in fact, because of the discount factor, the Q values are a little bit higher at this step because it's closer in time to the final reward. Okay, so if our neurons then, let me just show you how we did this physically on the screen. So what happened here in this control condition, we gave the monkey advanced information, so that's the valid cue. So the monkey knows from this point you have 100% probability of reward because this is 100% validity, and you're going to have to look up because this is the information I gave you. I gave you advanced information. But then I'm going to turn on this uninformative stimulus, and you're going to have to look at it. It gives you only noise. Nevertheless, you have to look at it. And then you're free to make the final decision based on the first cue, right? And now and now we're going to put this uninformative stimulus in the receptive field, and we're going to see if the neurons modulate to it. Okay. So remember, the prediction from the Q-learning model is that the neurons will show reward modulation for both informative and uninformative steps. But in fact, what LIP neurons do is they only modulate for informative steps over here. For the uninformative stimuli, the neurons respond because there's something in the receptive field and there's an eye movement going there. But they don't encode the different probabilities of reward that are associated with those stimuli. Okay? And this is very consistent. If we did a regression model in which we separated out the visual component shown in gray, and then the validity modulation for informative stimuli or the reward modulation for uninformative ones. And you can see that they're very consistently, the modulations are much higher for the informative stimuli. Okay. Okay, I see a lot of frowns in the audience.
No, just trying to
Yeah.
relate your result, which is
Yeah.
lovely, to blocking in a sense.
Yes. Okay. Excellent. Fantastic. Okay. Blocking. Yes.
Is that what you're going to do?
Yeah, that's what I'm going to do. Yes. So the question that that arises at this point, so blocking and reward prediction errors, right, is, well, okay, so this has shown that you cannot explain things simply by the cumulative future rewards from that emanate from a state, right? But what about reward prediction errors? Those are the key mechanism in reinforcement learning to detect when you get new information, right? Okay, so we did another control experiment to look for that. So how would reward prediction error work? Okay, so let's think again about our informative stimuli, right? 180 and 55%. Let's think what happens before those cues come on. The monkey is sitting at fixation. He's just waiting for something to happen. And he has a prior expectation based on the average of these three cues, right? So in this case, he would be close to about 80%, 78 to 80% of reward. So now, if he sees a black cue, 100% valid cue appear, that signals to him an increase in reward expectation. It's better than average. If he sees a green cue, that's about the same as average. If he sees a blue cue, that's lower than average. That's a negative prediction error, right? So could it be that this is, and then if you think again about the uninformative stimuli, of course those cues, when they appear, they don't change the reward expectation, so maybe that's why we don't get any neural effects. Okay, so we did a control experiment to see if reward prediction errors could explain the results, and what we had to do here was to set the monkey's expectation and then change that expectation in a controlled way, right? So the task was very similar to what we did so far. The monkey began by fixating, and he saw a cue of those three validities. And on most trials, the cue stayed on, the fixation point went off, and the monkey finished the trial just as I've shown you so far, based on this one cue. But on a critical fraction of trials, on about 20% of them, this cue stayed on for a while, but then it jumped and changed, right? So on this trial, for example, I told the monkey, oh, it's 100% valid cue, 100% reward probability. And then I say, oh, no, sorry, it's really 80%. I've changed it, right? And then I let the monkey complete the trial based on that informative cue. Right? So this is how we made a controlled change in reward expectation with a feint. And remember, these are only 20% of the trials. We can't look at all the combinations because we just would need too many trials. So we focused on a specific class where the second cue that appears, and this cue will appear in the receptive field, that's always 80% validity. But it follows the first cue can have 100% validity, or 80%, or 55%, right? So now we have a cue that appears in the receptive field that has a constant information gain, constant validity, but it's associated with a negative prediction error here, zero prediction error here, and a positive prediction error here. And the magnitudes of these prediction errors are very similar to the magnitudes that the monkey would see when they first see the cue, relative to the average. And so we expect that if the neurons really encode reward prediction errors, the responses to this one cue should vary according to what came before it. But we don't find that. So again, neurons responding, these are cues of different validities appearing in the receptive field. And those are cues of a constant validity with different reward prediction errors. And we don't see much of an effect. Okay, and here's just the quantitative analysis. You can see it's very consistent across cells, and across the two monkeys are shown up there in the insets with the two blue colors. Okay, so we don't have, yeah?
Just a question. I mean,
yeah.
Looking at the average firing rate of a population of neurons that probably you have sampled and selected in a particular way, and now when you say this information isn't in there, I don't know how convinced I really am, because you could train fancy classifier to look at the profile of firing of a large population of such cells, and you might find that you can read out lots of information from the population of these.
About reward, right. So that, sure, about reward prediction errors and cumulative future, absolutely. So, right, but what we're saying, but this difference here, nevertheless, tells you that there's something else. So I don't want to exclude the role of reward in modulating these cells. The argument that I want to make is that cumulative future rewards and reward prediction errors just cannot explain all of the responses. There must be some other factor at play, right? Okay, so this is how it summarizes it. It seems that the brain actually identifies, even when you have a sequence of actions where all the actions lead to a reward, the brain has some mechanism for identifying in advance when you will obtain information, right? And so I think about, for example, when you cross the street, there's a time when you make your decision, you look at the traffic light, boom, you've made your decision. Then you need to do other stuff to get to the other side, right? But that stuff you can sort of make offline with probably less attention. So what we're looking at here is a signal that says, hey, pay attention, you're going to get information, right? There's a cognitive recruitment that's going on at decision points. Okay, so informative from uninformative actions, even we can distinguish them even when they have equivalent rewards. And there's a preparatory response in advance of the information sample. That preparatory response is, of course, necessary to tell you where to move your eye, what to sample. And it may also be needed to prepare the brain to process the information. Right, so that's what I said. So that's it. Okay, thank you.
Questions?
Yeah.
This may be asking too much of a monkey. You don't need to train the monkeys that the up or down flow of motion should direct them, right? That they can learn themselves. So if you see the moving dots moving down, the monkeys will saccade down. No. That requires quite a bit of training, actually. There is some automatic tendency to congruence, motion congruent, but it's not. Let's train the monkeys for that. If you train them for that,
we train them for that.
Would you also need to train them to make the initial saccade to a cue that is potentially informative, or could they just learn that? And then presumably one of the things that they're doing is...
Yeah, that's a very good question. I think that they would learn to make the saccade to the cue on a as-need basis. And we have a little bit of evidence for this in this task. So in this task, we had to force the monkey to make the saccade to the cue always for our purposes. But we let them, we didn't constrain the amount of time that they spend looking at the motion. So after they made the eye movement, they were free to observe that motion for however much time they wanted. And the monkeys spend a lot more time looking at the informative motion and hardly any time looking at the uninformative motion, right? Now you can say, well, the uninformative motion was just noise. But we know from other studies that if the monkeys think that that's a valid cue, if you have random motion that's a valid cue, they actually spend more time on it if it's more noisy, so they try to discriminate it. Whereas in our case, the monkeys spend a lot less time when they knew it was uninformative. So I'm pretty sure that they will spontaneously make these sample, non-sample decisions.
This is something else that might already be in your data, but in the case of the monkey's initial fixation was to a 80% cue, 50%, sometimes they could be saying that. But say the 80% cue. Did they were they more likely to saccade and do the contrasted cue in the case where they could have done better? Maybe I already should, but if their initial fixation were on 100% cue, they don't need to look away and saccade to see what the other dot is. But if their initial fixation is on a green border, then it pays them to look and check out and see if there's a better cue out there.
Yeah, we didn't give them that option. So if the first saccade is what they got, right? After the first saccade, the motion started where their eye went. The other cue disappeared, I think. So that's it. We never let them change their mind.
Look at that initial
Right.
cue towards the crowd.
Right. Right. Yeah.
Is 20% sufficiently rare that it doesn't affect the response to the first stimulus? So if you compare monkeys in a condition where there isn't that 20% of the faint stimulus with a case where there is a 20% faint one,
Yeah.
could it be that what you call a prediction of 100% is actually for them something different, because they know that in 1 out of every 5
Yeah.
they are going to be told.
They're going to be
this is a
told.
lie.
Yeah, we worried about that too, and that's why we had to keep the feints rare. And
The question
right,
is 20%. You've
it's
checked-
20%. Well, we did check the behavior. Remember, 80% of the trials were just regular trials, and the behavior on those was no different than in the blocks where there were no feints. And also during their lifetime in the lab, the monkeys experienced a lot more no feint blocks than faint change of cue blocks. So we think that, yeah, yeah.
More question?
Okay, time for lunch. Okay, thank you.